on it. I'm on it. Welcome on in, everybody. Microphone is now on. Hope everyone's doing okay. It's been a very busy week here. Especially today. I've been going pretty much nonstop since I got on at 8, which is fine. It's good. A lot of good things happening right now. It's just one of those times where everything becomes busy, like everything kind of picks up a little steam. Content creation, we're starting that new type of content this week. So the little more balance between fun, practicality, and authenticity, which has been going well so far. I've enjoyed it. You guys seem to have enjoyed it too. Let me adjust this sound really quick. There we go. That's taken some time because it takes a lot more thought to make it more balanced. Because it's important I'm trying to make it a more balanced between authenticity, practicality, and fun. It's difficult hitting all of those facets while still trying to accomplish something that's valuable, and trying not to make it like the futon of content where I try to do so much that I actually accomplish nothing. We'll see how it continues to go. Wednesday was the first day that we really tried that out. It did all right. I think the ones coming up next week and later on will hit more of that, but so far so good. Everyone, like across all the platforms, it's been like a moderate response, which is kind of what I expected. If I'm trying to not specialize for individual social media platforms, I have to, so it'll kind of like TikTok. If something does well on TikTok, but not on Instagram, now I've kind of tried to make the posts a little bit more even, where they do marginally good on both. Who knows if that's actually a good thing or not? I don't know. Algorithms are algorithms. But yeah, that's been taking some time. Coral Solutions Hub, we had a review board meeting this week. A lot of submissions to that, so a lot of cool new stuff on the Coral Solutions Hub. A lot of new technologies. Great conversations with people this week too. So I love our community. That's why I continue to do work like this, because I like engaging with people in this community. They're hardworking, pretty down-to-earth folk. People know they take their work seriously, but they don't go crazy with it. I think that people manage their expectations and pretty well. I mean that as a compliment. Anyway, I'm also trying to continue working on publications. That keeps getting pushed back, which is not good. I need to just knock that off the to-do list, because that stuff needs to get done. Otherwise it's gonna get outdated. We got funding for it, so we owe it to NSF. We owe it to ourselves, my collaborators, and to just get it off the plate so I don't have to think about it anymore. So I'll have to find a way to fit that in, because it needs to happen. It needs to be prioritized. So that's what I've been kind of working on. Job list. This week was super busy. On the job list. We had 10. And I don't think I even got through everything, like all of my lists that I subscribe to and whatnot. Social media, direct info, LinkedIn, I don't think I got everywhere. And I still had 10 applications that I was able to find. It took me yesterday, I worked on it yesterday instead of this morning, because I had, the way my schedule worked, it made more sense to do the job list yesterday. It probably took me a good 3 hours or so, or at least 2. And then I also have to like spend some time identifying trends, because that's part of the extra content that we're doing, is like trends in the job market, which is great. I think that's smart. I think that's good. I think that's valuable to everybody. Just adjusting the lighting a touch. So yeah, very, very busy week. Very, very busy week. I don't like this. There we go. I'm happy with that. Probably a little too much lighting, but whatever. At least I don't look like I'm blending in the background anymore. So what I have on the docket today to wrap up our week here is I'm going to talk a little bit about reading papers, because that's a series that I want to start talking about on the for Reef Unity Network, is like strategies for reading papers, how it varies across career stage, break it down individually into like separate parts of a paper, so abstract, intro, methods, results, discussion, conclusions, all of that. I want to start doing that, and I need to get writing on it. I have not started writing them yet, which I need to do because it's kind of everything. I want to start it next week. And I still haven't written it yet. Because things have been crazy busy. I had something to add with that. So what I'm thinking with the plan, let's pull up the calendar. So I'll pull up our content calendar. Let's go ahead and take a look at this. Actually, what I need to do, I haven't started any of these. So I am not ahead anymore. I'm so not ahead that I haven't even highlighted that I haven't started making this content. So take that as you will. It happens. I'm not going to be ashamed of it. Not going to shame myself for not having it ready, because I got other stuff to do too. But we need to start doing it. Oh, I posted those, so we're good there. So that's all weeks. Normally I like to have all of this yellow by now. I like to have the whole month at least drafted, but I don't. So I don't know what you want to do about that. Let's see. Okay, yeah, so I wanted to start on Monday. Actually, I wanted the email to be how to read papers. What I think I'm going to do first is just an overview, kind of introducing the series, the points we're going to hit. Maybe I can talk about career stage. I don't know, that might be a high level thing I'd rather finish with. Yeah, I probably want to finish with that. But I want to do like an overview, and then I kind of want to do individual sections. So like I would spend a week in email, a post on the abstract, the intro, the methods, the results, discussion. Really, I could do it in one post, and we're going to read one today. We're going to read a blog post that breaks it down a little more, and they fit it all on one, so like technically I could do the same. But I want to break it down more. Otherwise I could just like copy paste, and I don't want to do that. I want a fresh take, or at least my take on each section. Whether you agree with it or not, it doesn't matter to me. Everyone, while there's like a general consensus on like how to treat papers, everyone still has a little bit of their unique strategy. So I guess I'm gonna use that as a segue into how I read papers. And I'm going to keep this in the context of grad student for now. And then I might discuss a little more on different career stages as we get further, see if it comes up naturally. Otherwise, I'll try and bring it up at the end. And then afterwards, we're going to review this blog post from Dr. Derek Miller. He runs this blog, Genius Gear Lab. It was a post I recently read. I liked. It was good. I liked it. On how to read papers quickly. I'm not so worried about reading them quickly. That's not really how that's not what I want to focus on. I want to take a deep dive into how I approach each section or as I'm reading to be effective. So that the blog post is quick and effective. I just want it to be effective because I don't read papers very fast. Because I need time to let the ideas kind of sit in my head and distill some of what they're actually talking about. So without further ado on that, let's jump into one of the papers I've written. We're just going to use one of the papers I've written as an example, and we'll cover kind of each topic. So as a grad student, if we're staying in the context of being a grad student, what I'm looking for in a paper could be a couple of different things. If I'm doing more of a literature review, kind of background, trying to build intro or build context for one of my studies, I'm basically going to be looking for general information that they talk about in the paper. Find other literature that can help me with additional background info. And then I also want to see if that study, what that study does, if I'm looking for different methods and their pros and cons. More often than not, I am trying to get the main conclusions from the paper, from the study that is shared. Doesn't always have to be the conclusions that the authors are drawn towards either. Especially if some papers tend to reach a little far with their conclusions. If I read a paper and I don't agree with their conclusions, I'm not going to cite their conclusions. I'll cite kind of my own. Not saying I'm making up answers, but there are data in the paper that you can probably draw reasonable conclusions from, that the authors do probably talk about, they might just not emphasize. So I'll pick some of the conclusions, maybe not all of them, and it may not be the main one for the paper either. So in the case of this paper that I wrote, I talk about high within colony growth variation and how it relates to genotype, so host and symbiote genotype and potential interactive effects. The main goal of this paper was to find interactive effects. What I ended up finding was high colony growth variation. That could be a conclusion. It's like this species combination, it was really difficult to find. And that's what we did find. It's with Acroporis cervicornis and Symbiodinium fiti, they kind of hold on to their partners. So it's kind of genotype specific. One host of cervicornis will more often than not, or at least in this study, we found harbors one genotype, maybe more than one genotype in separate ramets, in separate fragments, but more often not holds on to the same symbiont genotype. There's not a lot of mixing. So if I'm browsing the internet looking for papers, going to Google Scholar, typing in keywords, key questions that I might want to answer, so first thing I look for is the title. Does it seem like a good story that I want that closely resembles the study that I am trying to conduct or find literature on. So if I wanted to talk about interactive effects between host and symbiont genotype, this would be a great one. Phenotypic variation across either host or symbionts, this could be a good paper for. This title really highlights that for me. But it doesn't show that it's currently highlighted. Wait one sec here. The moment I started that, carried over. When I hop into a paper and the title looks good, interestingly, so most people will like quickly skim the abstract to see if it confirms like, hey, this would be a good paper. This is giving me some conclusions or methods that I'm interested in. I don't always skim the abstract though. When I'm reading papers, sometimes I will just skip right to the figures. See if they're going to tell, like you can tell a lot from the figures on what story is going to be portrayed by the types of figures that the authors share. So I might just quickly skim the axes. I'm like, okay, they're talking about colony growth rate. We have different genotypes listed here. That would be, you know, a quick tell for me. Like, all right, this is probably something I want to tell a story about. I did that a lot for my microbial studies, not so much this one, but a lot of my coral microbiome and ecosystem microbiome. There are certain figures like principal coordinate analyses I would look for and be like, this is the story. Yeah, they're probably talking about what I'm interested in it. Otherwise, I will read the abstract to see if they're talking about what I want. It depends on what I'm going for. It depends on if I'm looking for methods. It depends on if I'm looking for conclusions. How I approach that is a little weird. Most people just read the abstract though. I do know that. Most people stick with that. I think the blog we'll look at in a little bit talks about this more. The introduction. A lot of people will gloss over, especially if it's something like they're well versed in, they don't need the background information. A lot of people might skim this. If it's a new topic, people will spend more time on it. So if I was going to read a paper on coral metabolomics, which I know very little about, I'm probably going to read a little on that to get more context for the study and kind of what they're focusing on. If I'm doing this for a literature review, I tend to read the introduction in greater detail because it's going to provide references. References attached to other conclusions or previously like putative information that I need to reference in my own introductions or literature reviews. I need to know those original sources. So I will spend some time, if I'm doing a literature review, be like, hey, cool, so this one's talking about corals, or Cervicornis has fast growth rate, branching morphology, that's important. These studies talked about it originally, or were other literature reviews that reference it and talk about it in greater detail, had some conclusion involved with it. So I might flag some of these later, be like, hey, that's for future reading. You should either open the tab, download it now, or write a note, keep track of it some shape or form. That's what I do even if I'm not doing a literature review, I spend some time. You never know what interesting things people are going to reference and you can learn about. The key with introductions, though, is that I see this with a lot of early grad students. I was guilty of this when I first started. Do not cite a conclusion. It's not a conclusion, but do not cite information provided in the introductory text by the authors. So what I'm talking about with Cervicornis being having a fast growth rate and branching morphology, someone would not reference Gamache 2026. That would be inappropriate because I did not do that study. Who did it? Foster-Meinerson and Young and Schwartz. They were the authors who conducted that study, and they're the ones who are supposed to reference. So you do need to go in, read those studies, and potentially follow up on further literature from there. It didn't show. Hang on. That crashed really quickly. I hate this. There we go. Yeah, so fast growth rate, branching morphology. You can't cite Gamache 2020. You can't cite Gamache 2026 for that. I didn't do those studies. These authors did. These studies did. Or you can follow further literature through those ones, depending on how well I cited them, or if they were literature reviews themselves, which this one was. Regardless of the purpose of why I'm reading a paper for literature review methods or conclusions, there is always one thing I try to pay attention to in the introduction. And thankfully, most people keep it in the same place. I look for the purpose of the study. It is usually in the final paragraph of the introduction, and more often than not starts with here, or in this study, or something along those lines that sets up the purpose of the study and how they did it. This is almost like another form of an abstract to me. It's just without any, there might be a little bit of conclusions in there depending on the journal. Some authors might include it, but here's where I get all of the absolute context for everything that's going to follow. It's usually summarized in that last paragraph before the methods or results, depending on the journal. I want to know the purpose so that I can really focus on the methods to see if the methods reflect what their purpose was, and then see if that was relevant to the conclusions they eventually draw. So that's how I approach the intro. Long story short, I try to, if I need extra context, I'll read the whole thing. Otherwise, I'm looking for this purpose. I'm looking for the purpose of the study. All right, next section, we have the methods. I am inconsistent with how I read the methods usually. I will at least give them a skim. Again, to make sure that it fits the purpose of it. But I'll be honest, I don't always do that. I do sometimes just jump to the results after the introduction, the figures and results. At some point, I usually return to the methods, so if I get confused about some of the results, I'll return to the methods, be like, oh, okay, this is what they did, or they didn't clarify this, and I'll get a little bit skeptical, which is fine. You're supposed to be skeptical. That's science after all. But yeah, if you're approaching this in the context of a grad student, the method section is a great place to learn new methods, find new methods if you're starting a new project, see how others in the field have done it. You can confirm that your methodology for your project, is robust. People do it similarly in the field. I had a third point for that. I'm blanking on it. Oh, I mean, really, it's just, validate that the study is responsible, ethical. Methods do get treated very differently from journal to journal. I mean, everything. I could say this about every section. Every section is treated different from journal to journal. Some of them I feel like it's information overload, but to be repeatable, sometimes you do need that. I understand. Let's see. how do I want to break this down further? I don't know. How did that pass proofs? Oh, that's supposed to be capitalized. I think. It's a type 1 heading. Oh, how did I let that slip through the proofs? You always miss something. It's annoying. You always miss something. I know. I guess I bounce back and forth. A lot of what I probably do is I bounce back and forth between the methods and the results. Because I usually don't remember everything when I get to the results. I would say more often than not. More often than not, I probably go from the introduction, the purpose of the paper, to the results and discussion, mainly the results. If I were to like summarize how I do it, I probably do title, abstract, intro, results, methods, then discussion. Or I'll bounce between, because you don't have to read it chronologically from beginning to end. That's usually not the way to do it, because it's not very intentional. It's very easy to passively read and not pay attention if you start from the beginning and read it through to the very end. So my approach, which isn't everyone's, I know not everyone does this, is like, yeah, I'll go intro to get the context and the purpose, then I'll go to the results, I'll look at their figures, decipher them, make my own conclusions from the data, verify them with the methods, and then I finish with the discussion to see if my interpretations align with what they're talking about. So if I were to finish the intro, and I'm like, okay, what did this study do? We measured growth rates of genetically distinct Cervicornis colonies to assess interactive contributions from genotype opposed to symbiont. Then I would probably go down to the figures, the results, and see if that is present. If that information is there, can we see interactive effects between genotypes? And so we'll look at a figure. I'll start with the axes usually. So we're measuring colony growth rate in these ones. We have different genotypes listed on the bottom, host over here, symbiont over here. And then we have separate panels where we organize growth rates by host genotype. So this is Cervicornis host genotype, and all of the points here are going to be, or the shapes rather, are different symbiont genotypes. And then we have the reverse. So we have symbiont genotypes here, and then all of the points are different hosts. And it's showing some differences in growth rate, although there's statistics that I provided, not too much of a difference. Same thing for symbiont genotype. Although for context, I didn't actually run analyses on that because there was really no way to compare them rigorously. I'm like, okay, we at least measured growth rate and we see a high variation. That's how I would walk away from this figure, be like, we see variation across genotypes and within genotypes. So even with the same host and symbiont, we see variation in growth rate. Although across, there's not a lot of distinction, except at extreme ends. Same thing for symbiont, like different hosts. There's variation within the hosts. There's variation among the host genotypes, and then within a symbiont pairing as well. So variability, that's what I would take away from this. And then the next one, okay, we're looking at colony growth rate again with two of our symbiont genotypes. One of them has different host genotypes, while the other only has one instance. Did I run statistics on this? I forgot. I should have, except I can't because there's only 1 point. That's why I didn't. But ultimately, this is supposed to show like, oh, the only time we had an instance of a host genotype having multiple symbiont genotypes, it happened once across all of our coral and symbiont pairings. So we had one host that had multiple red, one host with multiple symbiont genotypes, one host genotype, multiple symbiont genotypes. Okay. So it's rare. In Florida cervicornis within this nursery. Those would be my takeaways. Like, okay, they didn't really see much. They saw a lot of variation, but they didn't have differences in multiple hosts with multiple symbiont genotypes. So you probably couldn't assess interactive effects between host and symbiont genotype pairings. And to cut to the chase, yes, that is what we said. And so I'm biased by that I do know the conclusions because I made those conclusions. So what I would do next is I'd probably go back to the methods just to verify. Like, okay, double check what they did, if it makes sense, especially like their statistical analyses, make sure those are robust. And then like how they actually did it. Again, I might even skim it before I go to the results. More often than not, I do it afterwards. Is if I have a good idea of what the study is. If it's something new, completely new to me, I'll spend more time on the methods. If I already kind of know the gist of it, especially with the microbial stuff, I'm like, yep, 16S, that is a pretty standard pipeline. I don't spend too much time on that unless I see something really weird. So after I finish with the results and the methods, I have a good, clear understanding of my conclusions of the data provided. Then I will read their discussion points and see if they are similar to what I concluded, if they fit the original purpose of the study, and to make sure that they didn't overreach on any of their conclusions. Easy to do sometimes. I've been guilty of it myself, and I've had to walk some things back. It's fine. We get excited. We get excited. It's new. It's important that people hold us back. That's why we do peer review. All that jazz. I already told you like the conclusions I make in this paper are the ones I was talking about with the figures, so I'm not going to spend too much time on that. Something else you might be looking for in the discussion are references. Again, they may not be exactly like they are in the introduction. There might be additional ones based on new conclusions that were made. You have to verify or reference or discuss conflicting pieces of evidence, and you pull in other studies for that. That's where I kind of pay attention to that, write down any papers that might be helpful for the future. It's important, I make sure it's important that I don't just like copy paste the discussion. I don't only read, like, I don't open a paper and only read the discussion because they may have made something that I don't know, a conclusion that I don't agree with, or the data might not support, or just not fit what I'm trying to talk about in my own work, like whatever the reason may be. That's why it's important to see the results match and align with it. There are, I've found that papers that do include conclusions, I like them to wrap up, but I definitely don't read them on their own because usually they're vague. They're kind of supposed to be, but I wouldn't take conclusions exactly from that. I'd rather draw them from the results and discussion. So that's a quick rundown of pretty much how I approach papers, reading them as a grad student. So this is how I would approach a paper as a grad student. Because really what you're trying to do as a grad student is identify like where and how other literature, other studies fit in with your work. Because you're trying to write your thesis, your dissertation, you need things to support your claims, you need things to weigh alternatives and alternative explanations. You're pretty much just trying to build together like this coherent thing. Now, if you are like a PI or long-term researcher, you're going to look at it with a little bit of different lens. You might be thinking more about research questions. So like, where's the field going and what projects can be done to help advance the field? You still will do that like as a grad student. You're trying to, stay on top. You're trying to make novel contributions to the field, but you're more just trying to help your own study get across the finish line. Later career researchers, like they have that understanding. They have multiple research products underway, so they might think a little bit further ahead. They're thinking about the rigor of the paper. They're thinking about future research questions, collaborations, places where they can, apply for new grants, new funding. So it's a little different how you approach it based on what career stage you are in. For the posts or the content pieces that I plan on writing, I'm probably going to stay within the lens of grad school. I think that will be more relevant to more people. I think they would be more useful to grad students than would be to tenured professors who know a lot of this already. Nevertheless, everyone can learn something new. And everyone does approach papers a little bit differently. I know my style is not. There are differences, and maybe we should read about those next, some of those differences. So I pulled up a blog post, and I'll do a little bit of back and forth with the author and see what I agree with, where I diverge, and maybe, provide some context on why I do it differently. But I've read this one before. I've read this article from Dr. Derek Miller before, and I do agree with it. I don't think he's there are things like, oh, that's totally wrong. It'll be just more like, oh, I do it differently, and this is why. So let's start here. Just in time, Anna. You got to the good part of the discussion. What I basically did, I walked through one of my papers. Mid-camp, but still want to say hi. Well, welcome on in. We just spent a little time going over, I walked through how I approach papers, using one of my own papers as an example. I thought that was easiest because I have the most context for it. Now what we're going to do is switch to a different blog who has talked about reading scientific papers, and we're going to see where we agree, see where we differ, and it's perfect that you're here for this because you can put in your opinions too and how you might differ from it. It's gonna be fun. So, okay. So he first starts, So what is your goal? Reading a paper. That is the most important thing. You shouldn't just read a paper because it's in your to-do list. You need an actual intention. You need a reason for opening that PDF and reading it, sitting down to read and take notes on it. One of your goals, getting a solid foundation in your field, doing a literature review, starting a new project. That's a way to do it. Collecting the newest research for cutting edge. Oh, we'll ignore the literature review there. Yeah, staying on top of your field. Seeing what else is being produced right now. And then finding ideas and inspiration to further your own research. Yep, this is what I kind of just mentioned about PIs. They might be doing this more. Researchers will be doing that more. This would be like new grad students. This would be grad students, maybe like, could be at the beginning or towards the end of their degree. And then that would be more researcher focused, kind of with their looking ahead in the field. Then it talks about first dozen papers will be a grind. Yep. It does seem daunting at first. It seems like, oh, you're never going to be able to understand it, but it does get easier. Author says that here. I agree. It does get easier. Oh, this is a funny point he makes. The template introduction. So that is super common in the choral field. Pretty much every paper you read is going to start. Mine, this one's a little bit different. I kind of, because that journal I posted in was much more specific to choral restoration, so I had to skip it, but nearly like every choral paper is like, coral reefs are degrading at an alarming rate due to climate change. Pretty straightforward with it. So you'll read that a million times, and it's fine. You'll start recognizing other papers you've read. Yep, that's totally normal. How you get... Academics aren't exactly known for concise writing. Yes, we're always told to write concisely and approachably. Yet we end up doing that in publications. We think we write concise, but then we don't. We start sentences with like, interestingly, and then follow it with something that's not interesting in the slightest. Guilty of that too. What you think is interesting, or you're just throwing that in as filler, like it's not. It's never worth putting that. Skimming paragraphs for high value verbs. Yep, you'll start doing that. Looking for like sequencing, monitoring, key methodology words. Here I told you when I'm looking for the purpose of a paper, I will look out for things like here, or in this study, or we did this. I'll be looking for that. Looking for lists, so I'll look for numbers. There are a lot of things like your brain, your eyes start recognizing a lot more quickly. So yeah, he's spot on with that. We don't read things with us. Yeah, we don't read really long things. You find the content and you quickly notate that. All right, so now he's gonna give the breakdown now, similar to what I just did. Let's go ahead and do that. Let me make sure we restart this so it doesn't lag. There we go. Okay, we're good. All right. So number one, briefly read the abstract. Yep, this is so you can quickly find claims to make sure that you are interested in the paper, that the paper holds some value for you, whatever your goal might be for literature review, getting that foundation, or if you're looking for like new cutting edge methods, this is where you could probably find that info. Okay, and here are the questions you can ask before you start the abstract. Is it applicable to what I need? Are the findings significant enough to help with your goal? Yep. What's the most interesting aspect of the paper? That could be objective, that could be subjective. Sometimes it's just cool to read things. You should be reading things beyond your research anyway, just so you know kind of what's going on. All right, now this is where we already start to diverge. The author, sorry, I just want to get this name, Dr. Miller. Dr. Miller suggests reading the conclusions next. So he does a little bit of a different approach than me. He will jump to the end, so the discussion or the conclusions, and try to find the claims that the paper makes. And it seems he wants to assess like how rigorous those claims are. Some of them you might be able to tell are really far-fetched right out of the gate. I sometimes that can be hard to tell sometimes because we write so objective sounding or very logically, where you can make something sound not outrageous, but it still is. So that's why I don't always jump to the conclusions, because sometimes they can be hard to tell if they are outrageous or not. But still, you might be able to get some red flags. I don't have any issue with that. Here's where, okay, there's some good logic here. By looking at the conclusions, you can kind of set the framework for, okay, what do they think they accomplished with this work? And what was their reasoning behind it? I don't like doing it this way. I don't want to know. I usually don't read the conclusions because I don't want to know what the authors are thinking, because that can spoil what critical analysis that I am doing. If I'm start looking at the figures and I already know what conclusions they've drawn, that might bias how I interpret the data. Alternatively, what I do is I read the purpose, which is included in nearly all introductions, right at the end of the introduction. That way I can know what they're trying to do without knowing what they think they accomplished. I think that gives me an objective way to look at the data without spoiling, like, oh yeah, I can see how they did that. I can see how that was interpreted that way. I don't want to know that. I don't want to know that. Again, I have nothing against doing that. I just don't like doing it myself. Okay, so after the conclusions, Dr. Miller then goes to the figures and digs through the results and discussion for more detail. So this is where he goes in and critically analyzes the data and draws conclusions. Some of the specifics he puts in here, like writing the questions and seeing if you can answer it with the data. That's great. I can do it the same way I do it too, by starting with the intro and then the results. You can do that too. Like, did they answer this question? Yes or no? You're essentially looking for answers in the paper. Good strategy if you're confused on a figure and need some important information, he puts that here. Control or Command F, find figure 3. That way, some figures, they can't include every information, all the information, or there might be other statistics that just don't fit in a figure, and you have to actually read the results text for that in the main body. And that's a good way to do it. That's a good way to do it. I do it. Command F's your best friend. Okay, this is something we agree on. So he's already done intro, results, discussion, and conclusions. Well, really, he did intro, conclusions, results, and discussion. And then he does the methods. I agree with this. Like, he starts it with methods is usually the most tedious and tiring. I don't want to. That's why we don't want to do it first. Yeah, I totally agree. You don't want to tire yourself out before you even get to the meat of the paper. That's like not what you want to do, because then you're not going to be as alert, you're not going to be paying attention, and you'll miss those points, and what was the point of reading the paper anyway? So that's cool. We agree on that. Save the methods until the end. I said I jump around sometimes. Sometimes I'll look back at the methods as I'm reading the results. I do that too. I'm sure he's not explicitly separating this either. But yeah, we both agree, like, save the methods. If you really need to dig into them, save it for after you make the conclusions. What does he suggest looking for in the methods? So paper showed a different result than another one, so the methods may cause a difference. Yep, sure. Everyone does things, even if it's the same pipeline, there are subtle differences because everyone's system is different. If you're in different labs and different settings, different research goals, yeah, you get different results. You're sure you want to include the paper and you want to be critical of the way they conducted their experiments. Hang on, let's spend a second on this one. You're sure you want to include the paper and you want to be critical of the way they conducted their experiments. Okay, so it's like you want to reference their work and be critical of it. Yeah, you need to make sure you know what this study did and how they did it before you can be critical of it. You have to have your ducks in a row before you start swinging punches. Alternatively, you want to replicate their work. You need to know how they do it so you can replicate it. And hopefully their methods are sufficient to help you accomplish that. This is really common. This is a really, really, really common one if you're like early on in a project. So like new grad students, you're probably going to be doing this. This is where you spend more time with the methods. Make sure to note anything unique, odd, or unexpected. This is hard to do if you're new. This is tricky to do because you're not sure what is right, what is wrong, or unexpected, because it looks like a lot of jargon. But the more time you spend in the field, the more papers you read, you start doing the work yourself, things start jumping out of the methods much more readily. You'll see different analyses, different R packages or Python packages, even in the like experimental procedures yourself, like the lab work or the field work, and you'll be like, oh, that is different. And you'll become more critical of it. You're like, oh, I know the flaws with that, or I know the benefits of that. I can understand why they chose that despite its flaws, blah, blah, blah. It's a lot easier to reason when you get a little more experienced. At first, it can be kind of weird, hard to take note of it, but you do get better at it with time. We'll come back to that figure at the end. So after the methods, summarize your thoughts and critiques. Absolutely. Absolutely. If you do not summarize your thoughts on the paper or the results from the paper, you're not going to remember that. There's no way you're going to remember that. You have to read hundreds of papers for a project. And summarizing helps your brain file and save that information. If you just finish the paper, final period on the final sentence, and you close that window, you close that PDF, how are you going to remember that? You need to write down the summary of your thoughts and critiques. Because you, 2 years down the line, or whenever you start writing it, which will always be a later date, you're not going to remember just by the title of the paper. Be like, oh yes, I remember this. You need to write in a little window, a sticky note in your citation manager. You need to write something to help you summarize the ideas. If you think you are going to use this paper extensively throughout your project, it might be helpful to consider the bullet points he describes here. What would you have done differently in the experiment? That's also good to know, especially if you have a defense coming up. You need to have an answer for what would you have done differently. You need to have an answer for that. Because there are a million ways you could have done it, and you need to know why you chose it that way, what its flaws were, and how it could be done better in the future. Is there an obvious gap or follow-up experiment? This is good practice for later on in your career, and also how you know how to address those gaps. When you include it in your manuscripts, it's helpful to know where the gaps or the follow-ups are so you can see where your work fits into it, its work, how it all integrates, and how it's advancing the field, how your work might be novel. And does the paper uniquely contribute to the field's body of knowledge? This is a good, just like mental checkbox. I mean, write it down, absolutely. Because some papers might not be helpful for you. Writing down like what its contribution is, very good to know. It's a quick way, it's almost like the abstract of the abstract. If you do that, like this paper, its main conclusion that will help with my project and my writing, this paper will address this issue. So like if I had to, let me pull this up. Let me pull this up again. If I were doing a study on interactive effects and I was going to be doing with Acroporis cervicornis genotype interactions, the contribution of this paper would be like, oh, Acroporis cervicornis like doesn't, like, host genotype and symbiote genotypes are pretty isolated. They kind of keep the same symbiont genotype. So it's good to know that, but we still see variation anyway. That was the contribution. Copy important figures into notes. So I would consider this supplemental work. I say supplemental, and the first line is, this is the most important step, but many don't do this. Exactly. Many people don't do this. I don't do it regularly either. It's important supplement. Supplement is always good. I spend a lot of time on the figures. So I don't necessarily write the important figures, but I write down the notes of the figures and what they showed me. So in a way I kind of do this, but not always, and not to this, in this format. So like adding a figure image, that's good for like quickly skimming through it. I was a little lazy with that. It might be helpful too if you're not sure how to present information on your type of data. So if you're like, oh, I don't know how to present genotypes and PCAs, save those figures from papers that are most relevant to your work, and then you'll have an idea of what they look like. That could be nice. That could be nice. And then, okay, pick important references. Yep. I'm not sure he's saying do this necessarily in this order, because I know I bounce around with this. So when I'm skimming through the intro, I'll do that. Discussion, I might not pick out references in my first read through, because I'm trying to get the conclusions when I'm reading that discussion the first time. If I do a second read through of the discussion, or like I do see something and it immediately stands out to me, I'm like, oh, I need to know that for later, then I'll pull it aside and be like, yeah, pick out four references, circle it, highlight it, pull up the links, save those PDFs. What I did later on in my grad school career is I had a folder on my computer with references, and I would make sure, like, I would build a queue, but I couldn't add to the queue unless I removed some. So I had to read some to add more to it. I know that sounds weird, but like it forced me to read papers. It kept me responsible, like, oh, I need to add these ones in, so I need to keep reading through. And then I would print one out like every single day and help clear through that queue. And that was really helpful for me at the end when I was like studying for my defense. That got me to read responsibly daily. That was super helpful. Almost like he has here, like a read on read. I did a version of that, and it kept me accountable. Clean up the metadata if you plan on studying this later. So if you're using a citation manager, when you download the PDF, when you download the reference, the metadata don't always carry over very well. So authors' names might get mixed up. Some might have initials while others don't. if there's certain HTML coding embedded in the file, the citation manager might not handle that, especially with species names. Citation managers don't handle HTML of italics very well, so it'll be like less than sign I, greater than sign on either side. And so, yeah, it is important to clean this up, because if you're doing it all right as you're trying to submit a paper before its deadline, you're going to make mistakes. It's not going to look good, and you'll have to spend more time later. So if you can stay on top of it, it's great. I'll be honest, I'm pretty lazy with this too. I do it usually before I'm submitting the paper. It's better if you can do it sooner. Okay, also mentions tagging for your metadata. Yep, I usually have folders, like I have categories. Like he has review papers, tech papers. I did categories like that too. And then take a break. Okay, so if you have to get through a ton of papers, give me 1 sec, let me refresh this. All right, if you have to review a ton of papers, take a break after each one. So like 10, 100, yeah, you can't do all of this reading at one time. One paper alone takes me usually about 45 minutes to an hour to read through the first time. That's a lot of time. That's a lot of energy. That's a lot of reading. That's a lot of attention. That's a lot of focus. You need to take a break from it. Let's see what he recommends. Yeah, couch or outside. You know, I played around with this a lot. How to actually read papers. Should you read them only on your computer? Should you print them out? Should you read them on a tablet? I mean, to each their own on how they do it. I did a lot of my work, a lot of my reading on computer. This is where my citation manager was housed. It was just easier because the PDF was right there. But towards the end of my PhD, I switched. I started printing them out. Like I told you, I had this queue. It kept me more accountable. But it was so nice to give my eyes and my body like a break from sitting at a desk on a laptop staring at a screen. That got tiresome. So what I ended up doing, let me grab it. I started printing all of them out, and I put them in this binder. So this was my, what did I call it? The title of my thesis up here, my dissertation. And I called it my dissertation study guide. Has a little coral on it. Weird branching coral. It looks like it's from Star Trek, actually. Crystalline entity for you nerds out there. I started printing them because then I could leave my computer. I could close my computer, give my eyes an actual break, and I could move away from a desk. I could go sit on the floor and stretch. I could lie down. I had a little bit more flexibility, and I could write. Really, it was for my eyes, and so I could like stretch better. I really liked that. I really liked doing that. So if you can, I would, and you're able to. It's something you can do. Print it out. Print it out. Start keeping a library. It makes you feel like an actual academic too when you start building a library with like all these papers in it. Like look at how thick this is. It's actually not even that many. It's only like a couple months worth of papers in here. But it'd be cool to have that library. Now I still keep like all of the papers I downloaded, they still end up in my citation manager, don't get me wrong. And I will read them, like especially if I'm writing, I'll pull up the PDFs and I'll put them there. That's totally fine. I do a little bit of both. But if I want to sit down and be intentional with my reading, I like to print it out, have a pen, circle references, give my eyes a break. Give a break from the computer. You spend enough time on it. It's nice to do something different and feel a little, have your coffee, your afternoon beverage of choice. Feels nice to be able to go sit on the couch or outside as you indulge in the scientific literature. All right, some bonus tips. Let's see what he says. I don't remember. It's been a while since I've read this. So pick your top few controversial, confusing, or interesting papers and ask a colleague for their thoughts. Oh, that could be fun. So we would do this a lot. Not so much we'd pick a person. These are the papers we'd choose for lab meeting. So we would pick them, the whole lab would read it, and we'd discuss it over an hour. I usually had coffee. All of our lab meetings were in the afternoon, so not everyone did. I usually had a little bit left from the morning, and that helped. Email the authors to ask a question or thank them for their contribution. You know, me a couple years ago would have hated that. I'd been like, no, that's so cringy, or they don't have time for it. But I've become like such a community and people person. Like, go for it. Go for it. Do it. If you have a critique, of course, be respectful. But if you have a question or like want to thank them, like you really helped me with this, let them know. They'd love to hear that. People love when you say their work helped them. Like, celebrate that together. It's great. Yeah, great way to make a connection. Connection is everything, especially in our field. It really is. So, yeah, they are busy, but they will love it. They will love it, surely. Reward yourself for every paper you read. That's the last one there. Sorry, ever since was my phone buzzing every time, ever since we've started up the LinkedIn page, like it's been going crazy. All right, reward yourself for every paper read. Small snack or short walk. Short walk, short walk, short walk, short walk. Do it. Take a short walk after that. Even if you're reading it on paper, like give your eyes a bit of a break, give your brain a break. Movement after you spend a lot of time thinking is one of the best ways to like consolidate memory. That and sleep, like taking a nap is also a great way to do that. I found, especially with myself, like movement while I'm thinking or after I had like a really tough meeting or a tough paper, movement helps me process things really well. That's why all of the weekly updates, when I was doing those videos, I was always walking, because walking helps me think. If you ever watch one of my presentations, if you ever come to any of my talks or presentations, I am moving the whole time. I might stop and pause. I'm not like running around the room or anything, but I like to move around, get to different parts of the audiences, and my brain like can work through what I want to say a lot better if I'm moving around. I've noticed like if I'm practicing presentations, especially, I'll start different parts of the room will be like in my memory of what part of the talk I'm at. So if I'm on like the left side of the screen, I'm on slide 2 talking about like coral microbiomes. Then I'll probably slowly walk around. By the time I get to slide 3, I'm on this side and I'm talking about climate change, something, I don't know. Probably start with climate change before coral microbiome, but whatever. But like movement is such a great way to process things. Well, this is a good topic too. How do you choose which papers to read next? Especially if you're doing a literature review, you might pull like 15 papers from a singular paper, and your to-do list, your reading list grows exponentially. Yeah, so you're downloading 15 new papers for every one paper read. How do you do that? Suggest citation trees and metrics. I don't pay attention to those. I probably should. To me, they're just internet points. It is important to know the importance of a paper, but every paper should have some merit. New topic, begin with relevant review papers. Yeah, that is solid advice. Start with the review papers, build that library of relevant and important ones that they have found important. But they also caution too many review papers leading to too many citations to follow. Also, it can be confusing on like, where's the original data? And that's the next point. That's funny. So it's been a while since I've read this, but there's still some agreement. Prioritize experimental papers with high citation errors. Yeah. You need to cite experimental papers. That's where the data comes from. You can do a little here and there from review papers, like follow their conclusions, but you have to show papers that did the original studies to do that too. That's where you're gonna, get the essence, the good stuff. Prominent authors in the field. Yeah, this one's a bit interesting. If you're just starting, that might be helpful, just so you kind of know who the players are in the field. I've noticed in the past couple years in the choral world, like, I don't do that anymore. I might like look for the senior author, because I mean a lot of the prominent authors, like, they're all senior authors now. We keep that at the end of the author list. So a lot of the time what you see is like grad student names, which will be new. So they might not be like the prominent name. And also, like, all these different prominent authors, they have different research focuses within choral restoration. So they all do a little bit differently. So I don't pay attention really to the authors anymore. All right, this one, after you've covered a lot of ground, start taking more chances on less After you've covered a lot of ground, start taking more chances on less established authors. Yeah, okay. I jumped the gun a little bit. But at least in the choral field, like, the less prominent authors are probably going to be first authors. The prominent ones are going to be senior authors. So, I don't know. All right, final takeaways here. We've got 15 minutes to wrap this up. Don't be a passive word for word reader. Yeah, don't read papers in order. You need to be intentional with how you read scientific papers. You have to go into the paper with a goal, verify that that paper is going to help you achieve that goal. Read the paper skeptically, make sure you're paying attention to see if they draw relevant conclusions that are supported by their data, And then you have to summarize. You have to file and save that information in your head somehow, in your citation managers, in your library, however you do that. Dr. Miller suggests reading abstract conclusion, figures, results, discussion, methods. I do it a little bit differently, my way, and I'm not saying this is wrong. We've already discussed like the validity of doing this order, and that's fine. A lot of people do it this way. That's fine. I do it abstract, intro, so I know the purpose of the paper, figures, results and methods, so I'll like bounce back and forth between figures, results, and methods. And then I'll finish with the discussion and conclusions. Dr. Miller here likes to do conclusions to see what kind of claims the authors were making. I do not do it that way because I don't want their conclusions to bias how I interpret the data. What I do is I look for the purpose of the paper in the introduction, highlight the points that the authors are saying they are going to try to make. Then I jump into the figures and results to see if I can come to if the data they present can answer the questions they propose. And then I will compare what I interpret from the data to what they claim in the discussion and see how they overlap. So I think that's the biggest difference between how I read papers and how a lot of people, including Dr. Derek Miller of Genius Lab Gear here, reads them. Everyone's got a little bit of different style. It probably differs by field and journal too. So like this journal or this author, can't remember his field. I know he lists it. He's a material scientist. So they might just do a little bit differently their papers and what they discuss. So that's probably the main point. It's probably the main difference there. All right. Figures are anchors. Yes, the figures and the results are everything. That's where you do a lot of your critical thinking and see how people do things in the field. Spend the most time on the figures. So I'd agree with that. Clean up the metadata and use a good tagging system. Yeah, be responsible. I'm bad. I'm lazy with this. Do what you can to do it. Set a daily goal. Absolutely. Absolutely, you should do that. At least one paper. I got really good at it at the end of my dissertation. Granted, I wasn't doing lab work, so I had like more time to do it. At least get a couple papers in a week. You can do it every other day. That's awesome. But don't overwhelm yourself. And that already froze again. No way. No way. Okay. So that's kind of a glimpse into what we're going to be talking about the next couple weeks. Email newsletter, and then social media. There we go. Get the words out of my mouth. Social media and the email content. We'll be talking a little bit more about reading papers. Probably follow a similar scheme to this. I'll do a quick overview. I'll talk about how I read papers, and then we'll do the compare contrast on like different strategies. I'll need to obviously read a little bit more, like one blog isn't enough, and I plan to do that. Probably over the next couple days, I'll be doing it over the coming weeks, just so I have a good background in my head, and then I can do like a really good comparison of like my strategy versus others. So that's the plan there. That's the plan there. I need to write, I need to read, I need to write. So I'm talking about the importance of reading papers and having a daily goal. I have been bad about that myself. So you gotta practice what you preach. And I have not been practicing what I preach, although I haven't preached it yet. Reading and writing tend to be one of the most inconsistent, irregular, but if you can find a good balance with it, a good routine, it really does carry you a long way. But I think we're gonna leave it there for today. I think we'll leave it there. That was a lot of talking. I didn't realize how much talking that was and how quickly that flew by. But yeah, expect more info on this coming up. I think that's really it. I just posted the job list right before the stream. Should go take a look at that. Share it with folks. Go share it with folks. Get people in the community. But other than that, I hope you all have a great rest of your Friday. You have a great weekend, and I'll catch you on the next one. Take care.